Yeah, so this is a paper that just came out in PLOS One with Alex Lowe as the first author. Essentially, what we did was we took a panel approach to using mass spectrometry to analyze a series of proteins related to the endo-lysosomal protein disposal pathway. So it's a fairly broad range of proteins, but essentially linked by the idea of lysosomal function in one way or another. It's a panel that's been looked at previously in other neurodegenerative diseases. And in Alzheimer's disease, for instance, you find that there are a number of these proteins that seem to be associated with a disease signal. In Huntington's disease, we and others have discovered a small number of biomarkers with great potential. So mutant Huntington is the disease-causing protein, and the level of that is of interest per se, and because it's the disease agent, but also measuring it can help to predict progression. Neurofilament light is the other sort of top-tier biomarker where if you measure it, you get a really good idea of what's likely to happen in terms of that patient's progression. And those are sort of deeply embedded in the clinical trial landscape. We take a view that we need to examine all possible biomarkers that have a reasonable chance of being linked or being capable of predicting progression. And the lysosomal pathway has been implicated in Huntington's disease and other neurodegenerative disorders in lots of different ways, both as a cause or as a consequence of neurodegeneration. So we broadly speaking, once we think something is a good idea to look at, we like to take a very agnostic and systematic approach and a very standardized, hierarchical statistical approach to analyzing potential biomarker candidates. So we used this. We identified from the literature a series of primary and secondary proteins of interest in the lysosomal system, and essentially quantified their levels in CSF in a cohort of 80 participants, which includes controls, pre-manifest HD, mutation carriers, and people with manifest HD. And essentially, the study was negative. We didn't find any analytes that were systematically altered in HD compared to controls, nor anything where the level in HD was significantly higher than in pre-manifest HD. We then undertook a principal component analysis, which is a much more I won't say hypothesis-free, let's say data-driven approach, where you basically allow whatever has the strongest signal to rise to the top of the data set, but in a mathematical way. And again, that didn't reveal that there was any particular candidate or group of candidates that had strong potential. We identified a number of proteins that changed together, but no particular high caliber biomarker candidates emerged from that. And in another exploratory analysis, we look at whether within the HD group, clinical severity has any strong associations with any particular biomarker or particular protein. Again, we didn't find anything particularly earth-shattering, but one thing I think that was of considerable interest was that the level of amyloid precursor protein or amyloid protein in general seemed to be quite strongly associated with clinical severity on a number of measures in a way that was surprisingly robust, considering that overall there wasn't much strong disease signal in the data set. So, from our point of view, essentially a negative biomarker study, but one that highlights amyloid or the amyloid family of proteins, which haven't really been implicated in HD before and certainly haven't been studied as potential biomarkers, but implicates them as potentially being of interest for further study. And of course, the study of amyloid species in CSF and other biofluids is widespread in Alzheimer's and other neurodegenerative diseases. So it gave us a nice sort of self-contained result, but also one that gives us a potential avenue for further study. Certainly, one way or another, it'll be interesting to look at that.